Okay,
Just a minute.
let's start.
Wait, wait, wait. Somehow I can't get out of this window. How come? Okay. Whew. Thank you. Okay, so we're very happy and honored to have Professor Zoltan Molnar of the Developmental Neuroscience at University of Oxford. And he's a longtime friend. We met many, many years ago. First, I think at MIT in Boston, and then we met on a course, remember? Where Giorgio Innocenti was there. We lost him, right?
Mm.
And it was a developmental course in Italy. And so it was really good to follow up Zoltan's career. Um, he is an MD from the Medical University at Zagreb, Hungary, and a DPhil at the University of Oxford. He has studied profoundly the thalamocortical development, but also migration, differentiation, circuit formation, and some studies in the human brain as well. And he is now a professor at the Department of Human Anatomy and Genetics at Oxford and associated to a lot of different European networks of research, which he maybe can tell us about. And he's also a member of the Cajal Club, right? We really miss these meetings. Anyway, I think he has received several awards and he is part of several academies of science in Europe. And it's a real pleasure to have him speak to us today. Thank you, Zoltan, for
Thank you very
accepting
much.
the
Thank,
invitation.
thank you very much, Cecilia. It's really a great pleasure to be here. And I have so many friends and collaborators in Brazil. It's nice to see some of the names and some of the faces here. So today I will be talking to you about the transient cortical circuits that match spontaneous and sensory-driven activity during development. But before I start, I just wanted to draw your attention to a very important anniversary, which we wanted to celebrate this year in Oxford. It's the 400th anniversary of the birth of the founder of clinical neurology, clinical neuroscience, Thomas Willis. I don't know whether you know that he was the person who gave the term neurologia or neurology to the word. So he was born exactly 400 years ago in this house. So actually last weekend I drove to this house. It's 40 minutes drive. It has an 11th century chimney, and probably the house was rebuilt around this. So this is the house where he was born. And then the family moved to Oxford, and eventually he ended up living in this street where he did most of his work, dissections and brain anatomy. So this is the street of the Merton Street in Oxford. You can see Corpus Christi College, and this is the Merton College Chapel. Merton is one of the oldest colleges in Oxford, um, uh, uh, 12th century, 13th century when it was established. This is the Porter's Lodge, the entrance to the college. And opposite, this is the house where Thomas Willis worked and lived and probably gave the term neurology, neurologia in 1664 when he published his famous book, The Cerebri Anatomy. So now you know where the word neurology is coming from. So this is where he practiced medicine. He dissected animals, dissected patients after they died, and uh, and he had an enormous contribution to neuroscience. So when he was studying the brains, on the left you can see a human brain, and on the right a sheep brain. he discovered that the human brain had larger proportions in cerebral cortex than a sheep brain. And also he noticed that the human brain had more sulci and gyri compared to the sheep brain. And of course, every medical student have to remember the name of Willis because of this arteriosus circle, which is called the circle of Willis. But he was the number 5 who described this structure, not even the first. Nevertheless, the illustrations from Christopher Wren were so good that his name is now associated to this arteriosus circle. But for me, it was a bigger discovery to suggest that the cerebrum is the primary seat of the rational soul in man and the sensitive soul in animals. It is the source of movements and ideas. So he associated the higher cognitive functions with the cerebral cortex in 1664. He also looked at patients. And on the right, you have a brain of a boy who had signs of intractable epilepsy and learning difficulties. And Willis noticed that in the family had similar kids from the same marriage. And he suggested that this is a condition which was inherited. And he also suggested that the cortical abnormality is responsible for these problems with the higher cognitive functions. So he was way ahead of his time because probably we are looking here at a polymicrogyria case, which is known to be associated with problems in neurogenesis and neuronal migration. And probably Willis was looking at a case like that, um, where you have more sulci and gyri, but slightly smaller due to these developmental abnormalities. So based on the comparative studies and also observing his patients, he suggested that the higher cognitive functions of the cerebral cortex um, are responsible for these conditions. And he was absolutely right. What I would like to do today is talk about the connectivity of the cerebral cortex. And one of the most important connectivity of the cerebral cortex is with the thalamus. As you know that the thalamus is a huge relay nucleus where most of the sensory input is relayed to the cortex through these nuclei, but also you have some thalamic nuclei, the so-called higher order thalamic nuclei, which don't receive primary sensory input. They relay cortical interactions via the thalamus. And I think Paco Klaska was talking to you about these, so you must be very familiar with these two types of thalamic nuclei. But just to recap this, you know that most of the sensory input is going through the thalamus to the cortex, and in return, you have output from the cortex to the thalamus, mostly from layer 5, which layer 5 is also leaving the skull and will innervate the basal pons and also the spinal cord. But you have collaterals to the thalamus and also layer 6, which has a major input to these thalamic cells. And as you know, the upper layers of the cerebral cortex, they don't project out from the cortex. They are involved in many other connectivity, and Roberto is in the audience, so I have to mention callosum, but I'm not going to talk about this today. So the lower layers are the ones which have these interactions with the thalamus. And Rodolfo Lindas suggested that you have much more interaction with the cortex, from the cortex to the thalamus than from the sensory periphery to the thalamus. It's almost as if the thalamus and the brain itself is listening to itself most of the time, and very occasionally you have some sensory input which is adjusting this state. So you have maybe 10 times more input from the cortex to the thalamus than from the sensory input. So I'm very interested in these cortico-thalamic projections, which regulate what kind of input can reach the cortex. And as I mentioned, these originate from layer 5, 6A, and I shall be talking about a layer today, which is called 6B, which is the remnant of the subplate. And I want to suggest that this cell layer has a very special function in the adult brain. So if you look at the thalamic nuclei, you can divide them up into two types. You have the first order thalamic nuclei, which receive direct sensory input, and then this input is relayed to the cortex. The higher order thalamic nuclei don't get direct sensory input. They receive their input from layer 5 and 6 from the cortex, and they relay it back to a different cortical area, therefore communicating between cortex and cortex via the thalamus. So there is a transthalamic cortico-cortical communication via these higher order thalamic nuclei. And these nuclei really enlarged during evolution in the brain. Think about the pulvinar. Now, so I'm very interested in to study these kind of interactions, and in particular this layer 6b, which has been ignored so far. And in mouse now we have all these Cree lines, which you can use to selectively monitor and modulate the function of these cells. So here I just show you three examples. One is the RBP4 Cree line, where you have a subpopulation of layer 5 cells labeled, and you can see that they target the higher order thalamic nuclei. They have large boutons, and repeated stimulations result in depression of the response. So this is through inotropic glutamate receptors. Then you have the layer 6A projections in the anti-SR1 Cree line. You can see that there is no selectivity. They innervate both higher and first order thalamic nuclei, and they have small boutons, and repeated stimulation will result in a facilitation, and this is mediated through metabotropic glutamate receptors. And this layer 6b, which is kind of like the remnant of the subplate cells, is yet another cell population, and you can access these cells, for instance, with the DRD1A Cree line, and they have a selective innervation pattern of the higher order thalamic nuclei. But we don't really know what the function is and whether they are drivers or modulators. So before we get to these interactions, quite a lot has to happen during brain development. So you have to form a neural plate, a tube, then the telencephalic vesicles, and then you have a neurogenesis. The telencephalic vesicles will grow, and they are enormous. There's a stunning neurogenesis, and eventually, when we are born, we have most of our neurons ready, and there's quite a lot of wiring happened already in the prenatal stages. So we can probably survive from the week of 24 weeks, even if we are premature. And you can see that most of the neurogenesis, gliogenesis, neuronal migration, axonal growth occurs prenatal. But the myelination and also the cortical thickness acquisition is a postnatal phenomenon. We reach the maximum thickness of a cortex around the age of 11, 12 years, and after that there is a decline in thickness in the cerebral cortex. So all this is happening before birth. And I just show you this video of the neurogenesis. So most of the neurogenesis happens in the ventricular, subventricular zone. And then when the cells are born, then they migrate out to the so-called primordial plexiform zone. So all these red cells are the first postmitotic cell layer. And then this layer will split into two parts, to the marginal zone and subplate, by the later arriving cortical plate cells, which will develop in an inside first, outside last fashion. So this is the pattern in all mammals, including human. You have these transient cells, zones, subplate and marginal zone, and then the cortical plate will develop in between them. And most of the cells are born here in the ventricular, subventricular zone. Now this cell layer, subplate, has been described many, many years ago. In fact, it was named by Ivica Kostovic. Um, um, and since then, all these groups made enormous contribution to the understanding of this cell layer, including Pashko Rakic, Ivica Kostovic, Carla Schatz, Donna Ferrero, Patrick McQuillan, Patrick Kennard, and Heiko Luhmann. So we know quite a bit about these cells, and they are very important for the development of the brain. They have very high content of proteoglycans. So I'm just showing here some human sections of stained for this proteoglycan. And this structure also have very high water content. So you can image it with MRI, even in prenatal stages. From these images are from Mary Rutherford. And this layer, the subplate layer, has also have all sorts of interactions from very early on while the cerebral cortex is still being generated. You have endocrine, paracrine, autocrine interactions, and also you have electric synapses and also silent and mature chemical synapses in this zone. And this is kind of like the center of the cortical development. So first I will be talking about the origins and early connections of these subplate neurons during development. So most of these subplate neurons are generated in the ventricular subventricular zone, and they just migrate out to the primordial plexiform zone, as I just showed you. The interneuron components are originating from the ganglionic eminences, and then they migrate tangentially to the subplate. And it was really recently when it was discovered by Juan de Carlos and us that some of the subplate cells actually, they originate from the rostro-medial telencephalic wall. So similarly to the Caja-Rechus cells, which come from these three zones adjacent to the telencephalic vesicles, in fact, some of the subplate cells, they also come from there. If you place a tracer in the rostro-medial telencephalic wall, you can also see the migration of these subplate cells into the cortex from this source from very early on, and they also show these markers for the subplate cells. So it was Navneet Vashishta and Fernando Garcia Moreno, when they were in my lab, who established that by looking at the clonal analysis of the subplate, that indeed some of the subplate cells, they go through the TBR2 positive intermediate progenitors, and 30% of the subplate is actually amplified up in these intermediate progenitors before they reach subplate. So this amplification was proposed only for the upper layers, and now we demonstrated that this is present in the lower layers as well. So once the subplate cells are born, then they immediately start to form connections with the thalamus, and the thalamocortical and cortico-thalamic projections start to grow out together. They meet at the internal capsule and they co-fasciculate as they reach the paleo-subpaleo boundary. The thalamic projections cross the paleo-subpaleo boundary by associating to the early corticofugal projections, and then the layer 5 projections reach the cerebral peduncle, the cortico-thalamic projections reach the thalamus, and then eventually the layer 5 projections will go to the basal pons and spinal cord. So this choreography is present in all mammals, including human. And if you have a displaced subplate neuron population, like in the reeler mouse or the shaking rat Kawasaki, then the thalamocortical projections will follow this altered position of the subplate cells. So instead of gathering below the cortical plate like in normal, in the reeler mouse, they shoot through the cortical plate, go to the super plate, they accumulate there, and then the thalamocortical projections will then descend from this compartment to the cortical plate. So they follow the same developmental algorithm, but in relation to the displaced subplate cells. Now, the reason why they can go through the cortical plate is because they have a very close association to these early cortico-thalamic projections, and they co-fasciculate, and this is how they reach the super plate by associating to these fibers in the hostile environment of the cortical plate. Now, we have better and better tools now to study selectively these early networks and early scaffolds. A long time ago, we used the Gali Tau EGFP mouse, where some of the lower layers were labeled, and you can see this co-fasciculation in the striatum and in the paleo-subpaleo boundary. You can see that the thalamic fibers make their way to the cortex through this association. If you have an altered position or topography of these early corticothalamic projections, then you also have an altered pathway how the thalamic projections reach the cortex. So for instance, in the EMX2 knockout mouse, the early cortico-thalamic projections descend to the ventral part of the paleo-subpaleo boundary. And when the thalamic projections grow out, they also descend there, and then they make their way up to the cortex. So we have demonstrated this altered topography a long time ago with Guillermina Lopez Bendito. It's almost 20 years ago. So these early scaffolds have very important role in the guidance of thalamocortical projections. Right after this, the subplate is involved in the transient subplate circuit formation. So as the thalamic projections arrive to the cortex, they start to form synapses within the subplate. And we demonstrated this in Keisuke Toyama's laboratory in the Kyoto Prefectural School of Medicine, where we use voltage-sensitive dyes in thalamocortical slices at various ages. And when you stimulate the thalamic cells, you have a long-lasting sustained depolarization pattern in the subplate from the moment the thalamic projections arrive to the cortex, suggesting that you have functional synapses. And this is very much the same even in human. So Sarah Bandiera is looking at this in my lab now, and she traced off thalamocortical projections in human brain, and you can see that the thalamic projections start to invade the subplate in this 17th gestational week human brain. But in addition to the subplate innervation, we also noticed that some thalamic fibers, they actually descend into the germinal zone, and they innervate the outer subventricular zone. Moreover, we have also seen some transneuronally labeled outer radial glia cells, which is a very interesting possibility that these thalamic projections have an interaction with these progenitors, perhaps regulating their divisions. Once the thalamic projections start to enter the cortical plate, then they can activate the cortical plate as well as the subplate. And I'm going to show you a newborn, then a 2 days old, and a 4 days old cortex. When we stimulate the thalamus, you can see this long-lasting sustained depolarization in the subplate and also in the striatum. So these squares flash with 10 microseconds on this very old video. We prepared, and you can see that very long-lasting sustained depolarization in the lower layers corresponding to mostly to subplate. So initially, when you activate the thalamus, it's the subplate which is activated, and later on, uh, when you compare a 2 days old and a 4 days old cortex, you can see the difference. I lined up these videos so they start at the same time, so we stimulate the thalamus at the same time, and have a look at the rapidity of the response in the four days old. You see the columnar response, and by the time it's almost finishing, this is when it's kicking in the 2 days old. So just 2 days can make a huge difference in the spatial temporal pattern of this activation. And this is the time when the barrel cortex is being formed. So you can see that the learning rules can be very different in an earlier stage and the later stage in the barrel cortex, and probably that also explains why we have plasticity for certain stages and not in others. So there are all sorts of changes occurring at these stages in the circuitry. And it was really Patrick Canod in Carlo Schatz's laboratory and then in his own laboratory who first suggested that the subplate neurons transmit the thalamocortical activity to the cortex before the thalamic projections would even enter the cortex. And then you have a transient circuit which is feeding these loops within the cortex. And then these loops are essential for the final targeting of the thalamic fibers. And after they establish connections, then the subplate will disappear. And it was Andre Marcus Smith in Simon Bott's laboratory. We co-supervised Andre with Simon, and who discovered that there is also a transient somatostatinergic input, and these cells receive thalamic input, which is then relayed to layer 4. So we have these transient networks, including subplate, uh, which is essential for the adult maturation of these circuits. So without subplate, you cannot really form these connections. Let me just show you how dynamic this system is. So again, we are looking at the Galli-Tau EGFP mouse, where subplate and some of the lower layer cortical plate cells are labeled with their neurites. And you can see that initially these dendrites, they reach the marginal zone. And then later on, you have some connections um, um, gathering within the barrels. We are in S1, in the primary somatosensory cortex, in the barrel field of the mouse. And then they congregate here within the barrels. And then at the end of the first postnatal week, they start to move out from the barrels into the septa, and there is this transition from inside the barrel to out into the septa at a particular stage of development. You can perhaps see this better when we have a look at this in flat mounts, in tangential sections. So initially they are in the barrel, and then they move to the septa. However, if I change the flow of the sensory information from the sensory periphery, I can alter this integration of the subplate into the barrel field. And if I remove a row of whiskers, then these neurites will stay inside this region much longer, suggesting that the dynamic integration of the subplate neurites in the barrel field is dependent and regulated by the sensory peripheral activity. So when I talk about this subplate network within the cortical plate, I would like you to think about a dynamic scaffold, which is assembled and disassembled, dependent on the sensory periphery. So how do cortico-thalamic projections respond to sensory loss? I showed you some of the thalamocortical, but now how about the cortico-thalamic projections? So I showed you that the thalamocortical projections accumulate below the cortical plate, and then they grow into the cortical plate a bit later. Whereas the cortico-thalamics, they wait outside the thalamus, and then they grow into the thalamus a bit later. So it was again discovered in the Galli-Tau EGFP mouse when we looked at the outgrowth of the cortico-thalamic projections. We noticed that the fibers reach to the thalamus very early before birth. But for instance, the dorsal lateral geniculate nucleus is not innervated by these projections for quite a while, not until around the end of the second postnatal week. So there is quite a lot of waiting before they reach and innervate these thalamic nuclei. So we wanted to see how enucleation, so removing the retina, will affect this ingrowth. And Eleanor Grant did these experiments and noticed that the early cortico-thalamic ingrowth was much faster if you remove the retina at birth. So there was less waiting outside. And this has been demonstrated in the in two lines, in the Galli-Tau EGFP mouse and also in the NTSR1 Cree line, where you have a broader population, including layer 6, you still have a faster ingrowth into the enucleated LGN. We know that it's the activity of the retina, because if we block the activity with epibutadine injections, then you have a very similar earlier ingrowth pattern in the LGN, where we inactivated the retinal fibers. We also looked at the transcriptome, the maturation of the transcriptome in these enucleated animals, and compared the transcriptome with the normally maturing LGN transcriptome. And we noticed that, in fact, the transcriptome showed more juvenile, early signs. So the transcriptomic maturation was delayed in these animals. So far, what I can tell you about after removal of the sensory periphery, that you have a faster ingrowth of the layer 6 projections into the first order thalamic nuclei, and you have a delayed maturation. So the transcriptome is more similar to younger DLGN after enucleation. But there's another interesting phenomenon which Eleanor discovered, and this is the cross hierarchical plasticity. So this cortical area, the layer 5 projections from primary visual cortex, they usually project to a higher order thalamic nucleus, like LP, pulvinar. And if you remove the sensory input from the first order thalamic nuclei, then these layer 5 projections now will project to this first order thalamic nuclei. Normally, they never do. So that's what Eleanor discovered in the RBP4 Cree line, where these layer 5 cell population projections come in red. And normally they just fasciculate through the DLGN, but in the enucleated DLGN, they started to form synapses within the DLGN, and they formed functional connectivity. We know that these projections, they don't come from S1 because we traced from S1 and also they come from V1. So there is a cross hierarchical plasticity. So the layer 5 is now innervating first order deafferented thalamic nuclei. And you can see that the thickness of the dorsal band of fibers is increased. And also you have the V-GLUT1 and TD tomato positive butons highly increased in the deafferented first order thalamic nucleus, the DIGN. So there is a plasticity between these cortico-thalamic projections, and we call this plasticity the cross-hierarchical cortico-thalamic plasticity. We also looked at what genes are associated with these changes, and Krija did a microarray on these many years ago, and then validated the expression of some of these genes which are associated with these changes. So this, we don't know what is the role of this cross-hierarchical corticothalamic plasticity, but if you remove the sensory input from a first order thalamic nucleus, then you have the layer 5 projections now target this first order thalamic nucleus, which normally they don't do. And we are also studying whether layer 6B have a has a similar in growth under such conditions. But again, I don't know what might be the functional relevance of these connections, if any. So after the development, the subplate neurons start to disappear. So if you look at the cortical plate thickness, as Lana Vassung did, who is now at Harvard, and you can see that there is an increase in cortical plate thickness, and then the subplate thickness is declining with the exception of here in the opercular cortices. Eventually the subplate will disappear. We also know how you see this here in higher magnification. The subplate cells disappear, and we know it from the work of Marla Luskin and Carla Schatz, where they did some very elegant birth dating in ferret cortex. You can see if you birth date subplate cells and layer 6 cells, and then you follow them after birth, a few months after birth, the subplate cells will disappear, whereas layer 6 will remain. So there's a preferential cell death in the subplate, and the same is true in the primate. Pashko Rakic and Ivica Kostovic demonstrated this, that you have subplate cells dilution. They end up in the interstitial white matter, but nevertheless, there is quite a bit of cell death of these early born subplate cells also in the primate. So in primate, you have a very thick layer, the subplate cells, and then you have these interstitial white matter cells. in the rodent, in the mouse, you have a compact cell layer left behind, which is called the layer 6B. In some primates, like in the galago, you can also have a compact layer 6B, so this is not a primate exclusive characteristic. But these interstitial white matter cells, they express very similar genes as we have seen in the mouse. And we, together with Paul Menger, who has an amazing brain collection in Johannesburg, we looked at the interstitial white matter cells in lots of different species, and uh, you know, in all these species, you have a low percentage, but very significant percentage of interstitial white matter cells. Some of them are NOS positive, some of them are carotene positive, and the percentages can vary even within the same animal in different cortical areas. But in general, 3% of your brain is interstitial white matter cells. So why should you be interested in these 3% of the cells? And I want to convince you that these 3% can be very relevant for some cognitive conditions. If it was Akbarian who, with Ted Jones initially and then later continued in his lab, who looked at these interstitial white matter cells in schizophrenia and described more interstitial white matter cells surviving, especially here in these frontal cortical areas. And he used several markers to demonstrate this. And I regard these cells as the shadows of the subplate, and perhaps more subplate cells survive because of the altered development. And we are very interested in the possibility, together with Heiko Luhmann and Patrick Canode, that these cells, which survived in the interstitial white matter cells, they did not follow the normal development. You have altered activity patterns in the developing brain. You have asynchronized activity, you have reduced activity, perhaps because of in utero infection, inflammation, or drug exposure, or hypoxia, ischemia. And then you end up with more cells um, left over in the interstitial white matter cells. And that cortical miswiring can cause neurological or psychiatric conditions. So that's why we are very interested in the connectivity of these cells and in normal and pathological conditions. So in the final stages of my talk, I would like to talk to you about what we are doing to find out more about the adult remnants of the subplate cells, the layer 6B cells. How do they integrate into the adult brain, and what might be the function of these cells be? So we started this many, many years ago when we were looking at the gene expression patterns in embryonic and early postnatal layer 6B and cortical and lower cortical plate cells. First, we started with microarray, and then we did transcriptomic analysis. And I want to draw your attention that we started all this before the Allen Brain Institute did the fantastic job on this. So we had a very good idea about the gene expressions in layer 6B. And we also pulled out some gene networks, especially Anna Horder-Zabadisson, who led many of these studies. And she identified gene networks within the layer 6B gene expression, which clearly indicated that some of these disease association was much stronger than chance would suggest. And this was the layer which had the strongest association to these cognitive abnormalities and conditions. And also, Anna also identified some of the genes which were associated with the surviving subplate cells. So these complexin 3, CTGF, NUR1, they are genes. If a layer 6B cell is expressing these genes, then they have a higher chance of survival to the adulthood. Most of these cells die between postnatal day 2 and 8 days. This is where the peak of the cell death is. And if you stain for complexin 3 or CTGF or NUR1, then you have less cell death in those cells than an average new and positive layer 6B cells. So now we have the tools to study and monitor and modulate these cells. And we can also look at the selective output input of these cells. And we can also look at the consequences of postnatal silencing of these cells. So the idea is that we use the special characteristics of these layer 6B cells to use viral labeling to trace their connections anterogradely and also look at the monosynaptic input to these cells using rabies virus tracing. So we identified DRD1A Cree as one of the best Cree expressing lines to study this subpopulation of layer 6B cells. And we used viral labeling to look at the connections. And this is how we discovered that they mostly project to the higher order thalamic nuclei. So if you just cross a DRD1A Cree line to a reporter line, then you can see that these projections from these cells, they mostly target the higher order thalamic nuclei, like PO, LP, uh, and they don't innervate the ventrobasal complex or the DLGM. And this already suggested to us that there may be a selective association to the higher order thalamic nuclei. Then Anna used Cree-dependent tracing from these DRD1A Cree lines because we suspect that they are very different from the Cree, for instance, from this NTSR1 Cree line. If you superimpose the projection patterns from the layer 6B and layer 6A, you see that the layer 6A mostly project to both first order and higher order, whereas the red is only in the higher order. And this was confirmed with Cree-dependent tracing with adeno-associated virus. And just to make a long story short, I just show you the reconstruction of these. So all cortical areas, they have some kind of an input to the higher order thalamic nucleus. Some of them are bilateral, and some of them will also reach the superior colliculus. And so there is a very extensive connectivity to the thalamus from this layer 6B cells. What is interesting is that they do form synapses in the higher order thalamic nuclei. So you have a co-localization of VGLUT1 and the virus labeled projections, but they never form side branches in the thalamic reticular nucleus. Layer 6A, they always have synapses in the TRN, but not layer 6B. And also layer 5 never had that. So we discovered a third type of cortico-thalamic projections in addition to 5 and 6A. We have a type of projections from 6B which is selectively targeting the higher order thalamic nuclei. They don't have collaterals to the thalamic reticular nucleus. And we now know that they also have small boutons. So let me show you that evidence. So it was Suiji Hayashi who traced these layer 6B projections. And here you see with immunoGOD. And they were always much smaller than, for instance, from the layer 5 projections. He also looked at the size of many others, like the layer 6A and layer 5. And it was surprising to see that layer 5 can also have some small boutons in addition to these giant boutons. So this model, the RBP4 Cree, which is normally associated to these large driver fibers, they also have some small boutons as well. So it's important to note that. So who is providing the input to these layer 6B cells? And for this, we used Ed Calloway's monosynaptic rabies tracing techniques, where you have a starter cells, where you have a Cree expressed. In this case, we used the DRD1A Cree and the CTGF Cree. Then we injected the receptor, which was expressed in a Cree-dependent manner. And then you inject the rabies virus, which will jump one synapse, and then you see the input. And here you can see the starter cells. Yellow, and then you can see the input is provided here from these layer 5 cells into these starter cells. And Florina reconstructed these in various brains, and you can see that this prefrontal cortical injection in the CTGF3 animals, it labeled this extensive network of intracortical input to these cells, which was also described by Tim Zolnick, but in addition, in Berlin. But in addition to this, we could also see some projections from the lateral hypothalamus into these Cre-positive cells. So I'm very excited about this finding, that there is a direct lateral hypothalamic input to these layer 6B cells because of several reasons. One is that it has been known in the literature from the work of Bayer and colleagues from Geneva that layer 6B is the only cell group which is sensitive to orexin. Orexin is a peptide produced in the lateral hypothalamus, and in the absence of orexin, you can have problems with the maintenance of arousal. So you can have cataplexy. And what we noticed is that if lateral hypothalamus had a direct input to this layer 6B, then some of these effects of orexin might be mediated to the cortex through the layer 6B cells. And in addition to this, it's also known that the higher order thalamic nuclei are also more sensitive to orexin than the first order thalamic nuclei. So we might have a network of these higher order thalamic connections and 6B, which are specifically sensitive for orexin. And we would like to test this, whether it is really 6B, which is mediating some of these cortical effects. So we wanted to see what's happening if we silence these layer 6B cells. How will this affect the behavior of the animals? So how can we silence a cell population in the brain? So we decided to reduce the presynaptic vesicle release in these layer 6B cells by knocking out SNAP25. SNAP25 is a protein which was discovered by Michael Wilson. Without SNAP25, you have no docking and regulated release of the synaptic vesicles. So in the SNAP25 knockout, you have vesicular release, but it's almost random. It's not associated to the action potentials. So in a way, if you remove SNAP25, you silence these cells. And that's what we did. And we looked at the development of the brain. So first we silenced it in layer 5 neurons in the RBP4 Cre line. And Kim Correll and Yolanda Disser looked at these silenced brains. And you can see that the connectivity is absolutely normal in these animals if you just look at the reported gene expression at 2 weeks after birth or 3 weeks after birth. Yolanda and Kim, they also looked at the myelination. The onset of myelination was normal. It started at the same time. There was some difference between layer 5 and layer 6 in the number of OLIG2 and CC1 positive cells in the motor cortex. So there is a cell type specific difference. Silencing layer 6 but not layer 5 impairs myelination, which is an interesting difference. But again, I have to emphasize that the onset of myelination was not altered. Then Shuichi Hayashi looked at the size of the boutons in the thalamus from these silenced layer 5 neurons, and Shuichi discovered that they were much smaller in the silenced animals. And when he looked at this in more detail, he also noticed that the specialized synapses, where you have the dendrites of the recipient cells in yellow, they protrude into these boutons, which are pink or magenta. You can see that this is not present in the silenced terminal. So they form synapses, but you don't have these specialized structures. And initially, all the connections are fine, the synapses are formed, the axons are myelinated, but later on, you have some reduction in the myelin, and also you have some degeneration of these fibers and eventually cell death. And when this happens, the microglia moves in, and Augusta noticed that this movement of microglia, how it is, how the signaling is occurring, and what time, and how layer 5 will receive more and more microglia as these neurons degenerate. And what is also very interesting, that Augusta discovered that this is all gender specific. So in a male and a female mouse, the congregation of this microglia is very different. And eventually, this will lead to cell death. And layer 5 is the most sensitive for this silencing, and they will die off. You can see that after 8 months, a large proportion of these silenced layer 5 cells will die. Layer 5 is the most sensitive. The pre-expression in these three lines, the layer 5, 6, and 6b, is roughly the same around birth. But then the onset of degeneration is different, and also the reduction of the exon terminals is also slightly different. And we didn't even observe any cell death in the layer 6b, whereas in the 5, you begin to see after um, a few months. The different cell types, they respond to the silencing differently. But what is the consequence of the behavior? So that's why we looked at the layer 6b silenced brains. So we had all these three lines silenced, and I'm going to show you some data on the layer 5 and also on the 6b silenced animals, some behavior. So first we looked at the layer simply because we had most more brains available like this, and it was done all in Vlad Vyazovsky's laboratory, who is an amazing sleep researcher. And when Tomoko, about 4 or 5 years ago, had a look at these layer 5 silenced brains, she noticed immediately that they sleep much less. As you know, the mouse, they usually sleep during the the light period, and then they are active during the um, um, active during the night, but they have some naps, so they sleep even in the night. And you look at this mutant, they have a reduced sleep during the day. They sleep 3 hours less than a normal animal. And you look at in the evening, they are so active, and they had a bit of sleep, but there was a huge difference. So there was a striking difference between the silenced and non-silenced animals. Um, Lucas, who has been working on this project the last 4 years, noticed that sleep deprivation is not affecting the layer 5 silenced animals the same way as the control animals. So uh, they cope very well with sleep deprivation. And you can see here the time course of slow wave activity after sleep deprivation in the conditional knockout and the control animals. So we also looked at the circadian rhythm, but there was no difference between the layer 5 conditional SNAP25 knockout and the control animals. We also gave them jet lag, no difference whatsoever after these changes. Then we proceeded to the layer 6b conditional knockouts, and we expected some kind of a abnormality in the arousal and perhaps also in the sleep. But we didn't receive anything like that when we reduced the presynaptic vesicle release in layer 6b, producing the layer 6b conditional knockout. And Kim and Louise looked at the locomotor activity. They looked at the circadian rhythms. They gave jet lag to these animals, and they also put some of these animals to constant dark. There was no noticeable difference whatsoever in these. However, when Tomoko and Elise looked at the sleep activity, we didn't see difference either. So there was no effect on the sleep looking at just by EEG. But there was a huge difference in anxiety related tasks. These layer 6B silence animals, they don't have fear. For instance, in the elevated plus maze, they go out to the open arm more than the wild type. They also spend more time in the light box, in the light dark box. And also, if you put them into this Y maze, where you block one arm and then they have a chance to get exposed to one arm, when you open up this novel arm, the layer 6B silenced animals, they go 50-50 to the novel and the other arm, whereas 80% of the animals would go from the wild type into the novel arm after such exposure. So there is a reduced novelty preference in the Y maze. So we are very interested in the possibility that some of the uh, well-known effects of orexin on fear and anxiety, they are mediated to the cortex through layer 6B. And we would like to study this further, how layer 6B is mediating these effects. So we know that there is no Cree activity in the amygdala. You have some scattered cells across here in the striatum, not in the thalamus. And also you can see that this is going into layer 6, then just to 6B. But so we are quite confident it is an effect on the cortex. So we want to see how this lateral hypothalamic orexinergic input is acting on the layer 6B. We want to know, first of all, whether these DRD1A3 layer 6B neurons are orexin sensitive or not. Now we know this from patch recordings. Tim Zolnik in Berlin has very strong evidence for that. Um, we are also very interested in doing in vivo recordings from layer 6B during state transitions, when the animal is falling asleep or waking up, to have a look at the activity. We also want to look at the local sleep pattern in localized 6B inactivation or stimulation. And we are also very interested in the human interstitial white matter cells, whether they have similar orexin sensitivity. Looking at transcriptomic single cell data, indeed they have, but we would also like to show that with direct recordings from human tissue. So I hope I convince you that the cell population, which is the remnant of the subplate, is a very interesting cell population which could have some involvement in some of these cognitive conditions. And that's why it's very important to understand their integration into the cortex. I showed you that these cells have various origins and they integrate into the developing cerebral cortex and thalamus in a dynamic fashion. You have these transient subplate circuits, which are essential for the development, and then they disappear. And I also showed you that there is a plasticity both on the thalamocortical and the cortico-thalamic level. They respond to sensory loss. I showed you also some evidence for cross-hierarchical cortico-thalamic plasticity that layer 5 is now innovating the first order thalamic nuclei. And these plasticities were not considered previously. And I also showed you that how the remnants of the subplate, the layer 6B, is integrating into these circuits. And through these very special connectivity and also sensitivity to lateral hypothalamic input, we believe that they might have an important role in the brain. So I'm very grateful to my past and present members of my laboratory, whom I tried to acknowledge during the talk. And I'm also very grateful to all the collaborators over the years and also the founders. And I'm very grateful for your attention.
Thank you. Great, great talk. Oh, there's João.
Wonderful, wonderful. Yeah, it was wonderful. Wonderful.
Thank you.
Thank you, Zoltan. Let's go for the questions. And so the first question is from João. So he's appearing here, so you
Yeah.
can make your own question.
Yes, I would love to be first because I will have to leave soon because of the child. And it was such a pleasure to see your talk, Zoltan. It's good to see you out of Facebook. Another context. It was wonderful. I have tons of questions, so I'll only do one of them. It's that thinking of higher order, do they have a temporal sequence of development consistent of being made later than the first order? It seems that. And I asked a specific image of your VB with the Golly animal that had small battlefields in the thalamus being seen, and they seem to change the inside-outside barrel pattern. I had a question for the other, but I'll leave to my colleagues and students and whatever. It was wonderful seeing you, Zoltan.
Thank you. I'm not that familiar with the birth dating studies on the development of the thalamus, but as you know, there are some clonal analysis papers on this, and it is now clear that the first order and high order thalamic nuclei and the thalamic reticular nucleus, they come from very different progenitors. So they come from very different sectors of the neuroepithelium, and they also have very different progenitors if you do clonal analysis on them. However, I think just from the birth dating, I don't think it was concluded that the first order or the higher order thalamic nuclei are generated first or second. I think they are co-generated, but I have to check this a bit more. You remember these Bayer and Altman papers on the thalamus, like 40, 60 pages each.
Yes.
And I must say, I always started reading them, but at the end I couldn't. But the data is available, and also now we have some birth dating and clonal analysis papers on these. But it's really exciting that evolution produced two sets of progenitors, which produce two functionally distinct thalamic nuclei. So I think that's very interesting that it is such a general feature of the thalamus to have first order and higher order thalamic nuclei both. And regarding the innovation from the cortico-thalamic, you are absolutely right. There is a huge delay in how they innovate the thalamus. You remember that Mrigan at MIT, he emphasized that layer 5 is the first cell group which is innervating the thalamus. And Francisco Klaska did some of this work when he was in Mriganka's lab. And they are absolutely right. They are the first ones who actually enter the thalamus. But the layer 6 and subplate projections approach the thalamus very early on, but they don't enter. And it was always a mystery, what do these early cortico-thalamic projections do? Uh, um, I think I had a lot of discussions with Ray Guillory about this, and we were entertaining the idea that perhaps they wait in the thalamic reticular nucleus. The thalamic reticular nucleus have very similar characteristics as subplate. They are also very early generated cells. You have preferential cell death, and it is a possibility that the PRN is the subplate of the thalamus for the cortico-thalamic projection. But this is not at all clear at the moment. But you are absolutely right. It would be very important to study this maturational gradient, how the layer 5, 6A, 6B approach the different thalamic nuclei.
Thank you.
Thanks.
Now, Roberto, do you want to make your question?
Yes.
Okay.
Thanks a lot, Zoltan, for the marvelous talk. I was just wondering if you have any idea on the the function, the possible function of transient thalamocortical projections. Do they prime later developing growing axons? Do they transfer any kind of information to the cortex in these animals?
So Roberto, you wanted the transient thalamocortical.
Right.
Transient. So almost all the thalamocorticals are transient because they initially they congregate in the subplate. So in a way, they are in position very early on, and then they, while they form the connections with the subplate, they congregate there, and then they make their way to the cortex. So in a way, there is a transient circuit, and what you probably know this literature much better than me, but the callosal projections do similar things. And in fact, there was a debate whether some callosal projections accumulate and they never enter the cortex. I don't know that literature that well as you do, but it's not known whether you have thalamic projections which go to the subplate, but they never go into the cortical plate. And that's, I don't think it has been ever resolved. So it would be an important question. That do we have some transient thalamocortical projections which only reach the subplate, but not the cortical plate? I don't know. And where do we stand in the callosum? Do we know whether you have callosal projections which never enter?
I don't know.
You don't know?
No.
Good, I feel better now.
Thank you. Now we move to Patricia's question. Can you make it, Patricia?
Yes, thank you so much. Zoltan was amazing. I was very curious about these white matter interstitial cells, the shadows of subplate, as you called. And because you also mentioned that some psychiatric brains, I don't know if it was schizophrenic brains, but they had actually an increased amount of those cells. And I was wondering, and then you mentioned that perhaps, infections could play a role in this phenotype. And I was wondering if it's known already, the function of these cells, the white matter interstitial cells, and if microglia could be involved in pruning these cells, if it's known, what is the steps of the pruning as well. Thank you.
So there are lots of different types of interstitial white matter cells, and people usually use NOS, NPY, somatostatin, carotinin, even just NUN to detect them. To the best of my knowledge, there is still no specific study on single cell transcriptomic analysis just on interstitial white matter cells, but that would be very important. There are some studies on subplate now, single cell studies, and that will be interesting to see how they cluster and how many types of these interstitial white matter cells we have. And once we have that data, then it will be also very interesting to see whether in certain brain developmental conditions we have certain subtypes in more abundance than others. And at the moment, we don't know that. And also, we don't know whether this is restricted to the frontal cortical areas or across the entire cortex. But this would be very important to know. So these cells have been included in almost all functions. Regulation of the vascular supply to the brain, regulation of ectopic activity, schizophrenia, autism. They were also implicated in periventricular leukomalacia, that if you have a hypoxic ischemic brain damage, some of these cells might be more susceptible for hypoxia. It was Donna Ferrero who suggested this with Patrick McQuillan, that these cells indeed, they get damaged preferentially if you have hypoxic ischemic brain damage, and then you can't finish the building work because you remove the scaffold, which would have been needed for that. So I think we still don't have subclass specific markers which we could use for very rigorous quantification of these subtypes in different areas and in different conditions. So but I think it's very close now to be able to address these questions. So we have many different types of white matter cells, many different cortical these questions. These are not numerous cells. These are, 3% of your brain, but they can make a big difference.
Thank you, Zoltan. Thank you, Patricia. So now, Rafael Linden, can you make your question?
Yes, Zoltan, nice to see you. Great talk.
Thank you.
Thank you very much. I wondered whether you looked at or found something related to any transient cortical projections to nuclei of the pretectal complex.
Pretectal?
Yeah.
There was subplate projection to the tectum, which we didn't know what these projections might do. We didn't pay specific attention to the pretectum, but we could go back to the data and have a look. Why do you think the pretectum might be interesting?
Well, it's a mystery, actually. All those, there are several visual field representations there, and there are nuclei that receive cortical projections, others do not. So I think it's an interesting place to look at. Although it's a lot more difficult because there's very little known, especially functional terms. I was just wondering whether, since it's so close, it's where
they do. 6B does project to the. So I must tell you that all these projections I showed you, they were in the adult. The viral tracing with the cre-dependent AAV tracing was in the adult. And I think it is very important that we redo these tracing in the developing brain. And indeed, Anna Horder-Sabodison has been comparing the projections of subplate with the claustrum. Some of the subplate cells have very similar gene expression to the claustrum, like NUR1, CTGF, complexin 3. They are also expressed in the claustrum. So, in fact, Luis Puelles came up with a suggestion that perhaps the subplate and the claustrum have common developmental origins. I'm skeptical about that. We have lots of good discussions with Luis, but the gene expression is supporting his idea. But I don't think that we have enough lineage tracing evidence for that particular statement. But the new experiment will tell. So what we are doing now is we are tracing the subplate early connectivity, and we compare it to the connectivity of the developing claustrum. And the idea is that the claustrum and the subplate might have very similar extensive connectivity, and the claustrum will maintain this very extensive connectivity, whereas the subplate will, most of the subplate cells will die, they are transient, and the claustrum will take over these functions. So that could be a very interesting hypothesis that whatever the claustrum is doing in the adult, the subplate is doing it during development.
Thank you. I have a question here, too, actually. So, Zoltan, what kind of information do you think this 6B thalamocortical projection conveys? Is this developmental, not necessarily an activity pattern, or is it something else? What do you think these cells are conveying?
So you are asking the 6B to the thalamus?
Right.
Yeah, okay. So my favorite hypothesis is that if you activate layer 6B, then you open up the transthalamic cortico-cortical communication via the specific projections to the higher order thalamic nuclei. So the idea is that 6B, when you activate this cell in a cell layer within a particular cortical region, then it would project to the higher order thalamic nuclei and would tell them that, hey guys, something interesting is going on here. Pay a bit more attention to the context of that stimulus. And then they open up some of these transthalamic cortical communications to pay more attention to the context. So it would be kind of an attention signal, which they could signal to the thalamus. And if you think about it, in autism and schizophrenia, this pathway could have a role in the pathology if you have too little or too much of these pathways to pay attention to detail. So we are testing this now. Elise Meyer in the laboratory with Vlad Vyazovsky, who is an amazing sleep researcher. What they are doing now is they silence layer 6B, and they give auditory stimuli to the mouse, and then they look at how they respond to this auditory stimulus when they are awake or when they sleep, or when you use the oddball paradigm, when you have an unusual sound, like ting, 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 ting. So our prediction is, if you silence layer 6B, then maybe the mouse will not pay so much attention to that oddball than in the normal. So that's the idea, but we don't know the results yet. Alice is just doing the analysis for this experiment.
Really interesting. Thank you. Let me get Eduardo to make his question. Eduardo.
Thank you, Zoltan. It was very nice talk. So my question is about the projections from the hypothalamus. Have you looked to the development of it? Does it relate to the maturation of sleep, for instance, the development of sleep spindles?
Very good question. Eduardo, the problem with all this tracing, and then I don't know how to conquer that, is they take a lot of time. So when you express the rabies receptor, okay, you can cross a flock-stopped receptor with the Cree line, and then you save a few days because you will express the receptor. But then for the rabies, you also need a few days. So we didn't invest too much into reducing this time. So most of our tracing is in the adult, and we don't know what's happening during development. But I think probably now the techniques were refined that we could go much earlier. So you are suggesting that maybe you have a more widespread lateral hypothalamic input to the developing brain than in the adult, which would be very interesting.
Yeah, right. And if it works if it's participating in the development of the cortico-thalamic circuit that generates sleep spindles, right?
But we were a bit disappointed looking at the sleep of the layer 6B silenced animals. There was not a major difference. I expected a much bigger difference. The layer 5 was like a dream. Vlad told me that over the last 20, 30 years, since he has been in sleep research, he has never seen such a strong phenotype of sleep when you silence layer 5. I expected that for the 6B, but there was no sleep phenotype for the 6B silencing, studying with just the normal activity patterns. And also, Lucas recorded from animals when they are falling asleep or when they are waking up. And he could always see some kind of an activity change in layer 5 but not in 6B. But 6B is very difficult to record from because it's such a thin layer, and unless you specifically go for that layer, you might miss it. So I still didn't give up. But Vlad and Lucas, they are into layer 5. So the idea is that yes, 6B might be still important in local activity patterns and also in some kind of a state control. And it's not only just through the endocrine function of the orexin, but also through direct lateral hypothalamic input to the 6B cells.
Thank you. Now, I was wondering if this signaling, this hypothalamic signaling to 6B is related to depression, because it's related to anxiety, right? But is there any evidence in that direction? Since it also could regulate sleep.
I have no evidence for that. Only anxiety. Anxiety phenotype was very, very strong. So how would you test that? You would
you would have to look at depressive behavior.
Mm-hmm. When they give up swimming or these kind of things,
or
anhedonia.
sucrose preference, or you have different kinds of depressive behaviors. I think that would be interesting.
We didn't look any of these. But the anxiety phenotype was unexpected and very strong.
Yeah, that's really
and it is known that orexin is involved in anxiety. So our working hypothesis is still that perhaps some of the cortical effects of orexin is mediated through layer 6B.
Roberto has a question.
Zoltan, I was curious about the larger number of interstitial cells in schizophrenics. Do they extend to the whole cortex, or are they localized in certain sectors, such as, for instance, the prefrontal cortex or something like that?
So I think Akbarian always studied the prefrontal cortex and the frontal cortex. So I don't know whether they looked at other cortical areas and they just didn't find it, or they just always automatically go to the frontal cortex. Henry Kennedy demonstrated that these interstitial white matter cells, they have an area specific distribution. So they used NADPH diaphorease and NOS, and they showed that in the monkey, you have an area specificity in the distribution. So it's not homogeneous. Some areas have more and some less. So you are absolutely right. It would be very important to show these differences. With Paul Menger, we looked at lots of different species, and there is a gradient in the brain. In the frontal cortical areas, you have more of these interstitial white matter cells. But I'm not an authority on the schizophrenic brains, but there must be a reason why Akbarian is always looking at the prefrontal cortex. So maybe this is specific for that region. And also, we don't know. So in the normal brain, you have more in the frontal areas, but we don't know why would you have more remaining in the schizophrenic brains. Is it because you have a cortical developmental abnormality, and as a normal process, you would remove these cells, but if you have a cortical developmental abnormality, maybe they cannot be removed? Or is it a primary defect of these subplate cells? Or is it a primary defect of the overlying cortex, or both? So we don't know the causal relationships why these cells survive in larger numbers. There was a recent paper in Frontiers where they looked at the DISC1 knockout, which is a model for schizophrenia in mouse, and they showed that they didn't touch the subplate cells, they just touched the DISC1, and then you had more subplate cells surviving. So it might be also the consequence of a abnormal cortical development that you cannot remove these cells from the interstitial white matter.
Thank you.
Claudia, do we have any questions on YouTube still, or?
No questions.
Okay. So Zoltan, I think we've gone through the questions. Thank you so much. It's really
Thank you very much for your hospitality, not just now, but over the years. You know, I have very fond memories, you know, when Roberto invited me almost 20 years ago. I don't know.
Oh, yeah.
I don't know, but I still remember, and very fond memories. And let's hope that we get through these difficult times and then we meet again. So in Oxford or in other parts of the world. Thank you for your attention and for your hospitality.